Hello, everyone. My name is Dova Levin, and I am the National Senior Director for Educational and Patient Programming. Today, we are having a webinar on the overview of clinical trial participation with Dr. Jonathan Hogan, who is the Senior Medical Director at Cabaletta Bio. During this webinar, we will provide an overview of clinical trial participation and review Cabaletta's CD19 chimeric antigen receptor CAR-T study for people with generalized myasthenia gravis and Cabaletta's MuSK-CAART study for people with MuSK-positive myasthenia gravis. Sorry. I'd like to thank our sponsors today for making the webinar series possible. Our national presenting sponsors, Alexion and argenx, our national collaborator, Amgen, and our national sponsors, UCB and Janssen. Okay, I'm going to stop sharing. You can go ahead, Dr. Hogan, and share your screen and start.
Great. Let me share my screen here. Can you see my screen?
I can.
And I'm on full screen now?
Yep.
Okay, great. Thank you, Dova, for the introduction, and thank you very much for the invitation to participate in this webinar today. I'm very excited to be here. Again, my name is Jonathan Hogan. I am Senior Medical Director at Cabaletta Bio. Here's my disclaimer slide. And to just introduce the Cabaletta Bio vision in the context of what we're going to be discussing today is to develop and launch the first curative targeted cellular therapy for patients with autoimmune diseases. And we'll be coming back to this theme because I think that cellular therapy, and in particular CAR T-cell therapy, really does move towards the potential for a potential cure for patients with autoimmune disease, given the data that's been generated in patients with blood cancers and in some patients with autoimmune diseases already. So we'll be coming back to this theme because we do think that CAR T-cell therapy does represent a real shift in the way that we're thinking about the potential for responses in patients with autoimmune diseases. But first, we're going to go through a brief overview of clinical trial and clinical trial participation. And I realize that there's a diverse audience that is on the webinar today and that will be viewing this subsequently. So just like to level set a little bit around clinical trials with an overview. So what is a clinical trial? The purpose of clinical trials is to collect data about the safety and efficacy meaning how well a drug works for the disease being studied, of an experimental drug. There are several steps and stages of approval in the clinical trials process before an experimental drug can be available to the public. Drug testing begins with extensive laboratory research that can last years. During this time, experiments in animals and human cells will occur. And if this initial laboratory research is successful, then researchers send the data to the Food and Drug Administration, or FDA for short, for clearance to continue research and testing in humans. So there are a few different phases that are discussed as far as the types of clinical trials that are conducted, from the first time a drug is studied in a human all the way to a drug registration and approval in the United States, as an example, and then even studies that are conducted after that. So the next two slides will be describing these different phases of studies. And again, these phases may be different than accelerated for rare diseases or diseases with a high unmet medical need, meaning diseases that really do not have many approved treatments or that the treatments are not that effective. So we'll start with phase 1 studies. Phase 1 studies assess the safety of an experimental drug and are the initial phase of testing. Usually these involve 20 to 100 patients or healthy volunteers, and they determine the effects of the experimental drug on humans, such as how it's absorbed, metabolized, and removed by the body. They investigate any side effects that occur as dose levels are increased, and they may collect some efficacy data. Again, that's how well the drug works on the disease being studied, if patients are included, to learn how well the drug works to control the disease being studied. Phase 2 studies test the safety and efficacy of an experimental drug. This can also often involve several hundreds of patients, depending on the disease being studied. These are often randomized trials where one group of patients receive the experimental drug, while a second control group receives a standard treatment or a placebo, which is an inactive product. And these phase 2 trials may be blinded, and that means that neither the patients nor the researchers know who has received the experimental drug, which may provide information about the relative safety and efficacy of the experimental drug without introducing bias from either the patients or the providers or researchers knowing which drug the patients received. You'll see in a little bit that I'll be describing a phase 1-2 study that Cabaletta is conducting. And phase 1-2 studies are a combination of the principles I've laid out in this slide with phase 1 and phase 2 studies. And these occur when patients with the disease are part of the initial safety testing. And cell therapy trials also tend to be much smaller than trials with other types of therapies. So again, in the context of thinking about CAR T-cell therapy as an example of what's called a cellular therapy, a lot of times these phase 1 or phase 1-2 studies are much smaller than what I've described on this slide. Phase 3 studies confirm the safety and efficacy of the experimental drug. These are usually randomized or blinded. They may involve several hundred to several thousand patients, depending on the disease that's being studied. They provide a thorough understanding of the effectiveness of the experimental drug and the range of possible side effects. And if successful, they allow for the drug developer to request the FDA to approve the drug for marketing. So these are a lot of the ones that you may hear about in the news when a drug is therefore approved and then subsequently coming to market. In the lay press, the phase 3 study often makes a big splash because that's what will lead to the request for marketing the drug. And phase 4 studies are conducted after a drug has been approved. The objectives of those types of studies would be to compare a drug with other drugs already in the market, or to monitor a drug's long-term effectiveness and impact on a patient's quality of life, or to determine the cost effectiveness of a drug therapy relative to other traditional and new therapies. So this is just a brief overview. Drug development is a very complicated subject, but this is just a brief overview of the different phases of trials that you may hear about if you are a patient or provider or working within if you're a researcher. Who can participate in a clinical trial? Well, among other requirements, such as patients being given full information about the potential risks of the trial and patients providing informed consent, all clinical trials have requirements about who can participate. Before joining a clinical trial, volunteers must qualify for the study. The requirements that allow participation in a trial are called inclusion criteria. These are specific criteria that a patient must meet in order to be eligible to be included in the study. The requirements that disallow participation are called exclusion criteria. And these are a list of criteria that if a patient meets that criteria, they are not eligible. They are excluded from the trial. These criteria can include age, gender, the type, stage, and severity of disease, previous treatment history, and other medical conditions. Inclusion and exclusion criteria are used to identify appropriate participants, promote patient safety, and ensure that researchers learn from the information they need. Again, you can imagine how important it is to define the appropriate patient population that one wants to study in the context of a clinical trial to give the information that is needed. And just by changing any one of those inclusion or exclusion criteria, you can really impact, both in a positive or sometimes in a negative way, the data that is generated and the impact that that will have on the availability of that drug for those patients in the future. Some questions to ask before choosing to participate in a clinical trial. How long will the trial last? It can be from days or weeks or months or even years. Where is the trial being conducted? I think one thing that is very clear and very important to recognize is that geography is a very important determinant of not only healthcare and healthcare access for many patients, but certainly for the ability to participate in a clinical trial. So where is the trial being conducted? What treatments will be used and how? What is the main purpose of the trial? How will patient safety be monitored? Are there any risks involved? What are the possible benefits? What are the alternative treatments? Who is sponsoring the trial? Do I have to pay for any part of the trial? What happens if I am harmed during the trial? And can I opt to remain on this treatment even after the study ends? So again, you can always stop participation at any time for any reason. That is your right as the patient participating in the clinical trial. But again, these are just a list of things that you can ask about if you're interested in discussing with your provider, if you're a patient considering a clinical trial. Again, and there are strategies for addressing many of these questions. For example, when I mentioned things about geographic constraints as far as where the trial is being conducted, many trials do offer support for patients and their caregivers in terms of travel to the study site and other supportive measures that can be arranged through the clinical trial. So now that we've gotten through a brief overview of clinical trials, I'm going to open up the first poll question. This is for any patients who may be on the call right now in the webinar. What would you consider to be a major barrier to participating in a clinical trial for myasthenia gravis? 1, my inconvenience to work and/or family life. 2, financial considerations, that would be travel to or from the study site, missed time at work. Number 3, risk of experimental therapy. And 4, there are already enough safe and effective treatments for myasthenia gravis. I'll give a second for people to chime in on their answers here. All right, I think we have our results here. So it looks like about 60%, 59% of patients have chosen number 2, financial considerations, travel to or from study site or missed time at work. About 29% have chosen number 1 and number 3, inconvenience to work and or family life and risk of experimental therapy. And 6% have said there is enough safe and effective treatments for myasthenia gravis. So thank you for that input. And this is very important, right? I think that it's sometimes not considered enough in initial discussions, and it should be at the forefront of our mind around the burden on one's life, participating in clinical trials and making sure that that's a recognized aspect as we as a sponsor and as study teams and your providers think about your participation in clinical trials. So thank you for sharing your feedback on that. So again, I'm going to go back now to the vision statement from Cabaletta Bio, which is to develop and launch the first curative targeted cellular therapies for patients with autoimmune diseases. And now I'm going to shift gears and talk about CAR T-cell therapies for autoimmune disease. But first, I'm going to start with the story a little bit of the history of CAR T-cells for patients with hematologic malignancies, or also known as blood cancers. So the CAR T-cell therapy, we can see here, this CAR stands for chimeric antigen receptor. And I'm going to describe that in just a second. And these CAR T-cell therapies, there are 6 of them that have already been approved in the United States for certain blood cancers. All of the approved therapies are called autologous CAR T-cell therapies. Auto meaning that they're coming from the patient's own cells. So these therapies are actually made from a patient's own cells. They're engineered from a patient's own cells. So a patient undergoes a procedure called leukapheresis. That's where blood is removed from the body and run through a machine to remove some of the white blood cells, which are part of the immune system, from the bloodstream. And then the rest of the blood is returned to the body. Those T cells, which are those white blood cells, are then modified using a technology, a gene editing technology, to insert a specific gene or code into those T cells. This is done in a laboratory setting. And in this setting of CAR T cells, the gene that's encoded is coding for this chimeric antigen receptor, or CAR. I'll get into the target in just a second. Those cells are then grown outside of the body into the millions or even billions of cells and then reinfused back into the patient. So again, this is that autologous, auto, meaning coming from the own patient's cells. And the first approved CAR T cell was called a CD19 CAR T cell. And that CD19 CAR T cell targets a CD19 protein on cells in the body. And the reason that that's chosen is because the cancerous cells that were initially chosen for the initial therapies are B cell cancers that are in the blood, and B cells, which are also part of the immune system, all have this CD19 protein that one would want to target in the case of B cell cancers. So again, the chimeric antigen receptor here is targeting the specific protein that you want to target for killing those cells and getting rid of the disease that you're trying to impact. And indeed, if you look at the initial studies for patients with a specific type of leukemia called B-cell ALL that was approved in children and young adults up to age 25 for the first approved CAR T-cell therapy, which is called Kymriah, that patients who had been refractory to all other therapies previously and very bad prognosis in that setting, that those patients with one dose of this therapy achieved between a 53 and 81% complete remission rate with one dose of the therapy, and that between 40 and 55% based on these studies of those patients went into long-term remission. So this was really, really sizable and transformative impact and result for some patients with these blood cancers. It doesn't work in everybody, but for those patients that it does work in, having these long-term remissions with just one dose of a therapy has really been transformative. And this is where those long-term remissions had led to the idea of a theoretical cure for some of these patients. The poster child, quite literally, for Kymriah, the first approved CAR T-cell therapy, is a young woman named Emily Whitehead. Emily Whitehead is the first pediatric patient treated at the Children's Hospital of Philadelphia with Kymriah for that leukemia, B-cell ALL. And by last report that I'm aware of, she's about 13 years out from her therapy, still in complete remission after one dose of that therapy. And actually, she's a freshman at the University of Pennsylvania right now. And so it's really one of those true success stories of a disease that was pretty much universally fatal, with one dose of therapy, having a long-term remission with just one dose of a therapy. This is just a list of the 6 approved CAR T-cell therapies in the United States. You can see that there are 6 of them that target that CD19 protein, which is again present on B cells, and those are responsible for B cell cancers. And then there's another protein called BCMA, and that's a protein that's present on another type of blood cells called plasma cells. Those are another cells that are in the immune system, and those are the cells that when they divide abnormally and cause cancer, that cancer is called multiple myeloma. And you can see here there are 2 approved CAR T cells for multiple myeloma targeting BCMA. And now I'm going to switch gears after that, after I've set the stage with a little bit of information about the history of CAR T-cell therapy in hematologic malignancies or blood cancers, and talk about Cabaletta's CAR T19 molecule called CABA-201, which we are studying for patients with autoimmune diseases, including generalized myasthenia gravis. So the enthusiasm around using the approach of targeting CD19 with CAR T cells in patients with autoimmune disease has come from data from academic centers, that is university hospitals, that has shown that there's been this sort of resetting of parts of the immune system after CAR T cell therapy that's led to really promising clinical responses in 15 patients across several different autoimmune diseases, with a CAR T cell that contains a specific component called 41BB. And that's an important component of the CAR T cell that leads to its being really effective both in blood cancers and likely in autoimmune disease as well. So in these patients who either had, this is abbreviations, either lupus or a muscle condition called myositis or another condition called systemic sclerosis, 100% of those patients had a clinical response. With T cell expansion, meaning that the T cells that were given, they expanded and divided in the body like they're supposed to do. And they depleted the B cells like they're supposed to do because they're targeting those B cells by targeting CD19 within the first month. And that enabled robust clinical improvement by 3 months. With regards to safety, and I'll be speaking to this in a few slides more specifically, the rate of cytokine release syndrome, more severe than fever, is less than 7%, 1 out of the 15 patients. And I'll be going into this a little bit more detail shortly, but cytokine release syndrome is one of the adverse events or side effects that people worry about most in patients receiving CAR T-cell therapy. These responses that patients had were also very durable, meaning that they have lasted a very long time after just 1 dose of that therapy. So the longest follow-up in the first treated patient, as of the group in Germany who has reported these cases, it was up to 28 months of follow-up with no relapses occurring in any of the 15 patients reported by this group in Germany. And all these patients remained off of all other immunosuppressive agents. What this means is that with just 1 dose of therapy, that what this group has observed is that these patients have had durable clinical responses, and those have been drug-free responses. And that means that as of the time that they got the 1 dose of this therapy, those patients were off all other medicines that would be suppressing their immune system after about 3 months. And you can see here that there was also some information about the B cells, those cells that were targeted by the CAR T-cell therapies and what happened to them. And you can see here that the patients did have depletion of the B cells, right? These CAR T-cells are supposed to kill these B cells, but then the B cells recovered in all of the patients. And this is very important because if your B cells are depleted and killed for a very long time, then one can be at risk for the complications of that, including infections, because B cells help to fight infections. But indeed, we did see in these data from the German group that the B cells did come back in all of the patients so far that were studied, except for the last 3 patients treated. We're still waiting for the data on those patients treated. But for the first 12 patients, all of the B cells have been repleted. In those patients, they've come back and measured in the bloodstream. And again, in patients with more than 3 months of follow-up, all of them have had those B cells come back, followed by return of healthy B cells within a median of 4 months. And the interesting part about that, and this is maybe for those who are more familiar with the science, is that the types of B cells that came back are of a naive type of B cells, meaning B cells that are not as mature. There's lots of different types of B cells. And that's why there's been this idea that there's been a resetting of the immune system, at least with regards to the B cells, because the B cells that come back are different than the B cells that were there before this therapy was given. So this is very important data. And here's the group. This is Professor Schett. He is the rheumatologist who has led these studies in Germany. And again, he showed that it was feasible to conduct this type of trial in patients with autoimmune disease. He showed rapid and sustained disappearance of B cells, and this is in the lupus patients with the antibodies that are known to be involved in their disease process, that these antibodies were cleared, that there was cessation of lupus-associated manifestations, as well as complete withdrawal of all the other immunosuppression, that there was reoccurrence of B cells that was observed in all patients. This is the first 5 patients that were treated. And that the B cells coming back was not associated with the disease relapsing and coming back. And that, again, there's that resetting of the B cell system, which is associated with that drug-free response. And here's a picture of the first 5 patients. And here's a photo of Professor Schett, his colleague in hematology, Andreas Mackensen, and this is the first patient treated in Germany, who is now out to about 28 months after therapy, still in disease remission, per the last report from Professor Schett's group. So what is the rationale for thinking about this type of study for patients with myasthenia gravis? Well, we know that there's no curative treatments for myasthenia gravis, just like pretty much all the other autoimmune disease. There are no cures. B cell depletion, meaning that when you get rid of B cells with medications like anti-CD20 monoclonal antibody therapies, with an example being rituximab, has been shown to be very effective in some patients with myasthenia gravis. Not all, but some patients with myasthenia gravis. And other strategies to deplete the antibodies that cause or contribute to myasthenia gravis, like AChR antibodies or MuSK antibodies or LRP4 antibodies, using FcRn receptor inhibitors, which is approved for AChR positive disease, are approved for myasthenia gravis. However, available therapeutic options for myasthenia gravis have limited effectiveness, focus solely on specific symptoms, and are associated with serious long-term side effects. CABA-201, the molecule that Cabaletta Bio has, is an autologous T-cell product that is genetically engineered to target B cells via the expression of a CD19 specific chimeric antigen receptor, as I already described, and it also contains this 4-1BB CD3 zeta intracellular co-stimulatory domain and T-cell activating domains. And again, this is for more people who are familiar with the structure of these CAR T-cell molecules, but this is very important as far as how well these CAR T-cells work and how safe they are. Deep depletion of B cells in myasthenia gravis may lead to cessation of disease by removing a central driver of inflammation. That's those B cells that are making antibodies that are attacking the body, and allow the immune system to return to a tolerant state, resulting in deep and durable remissions off therapy. That's off of all other therapies. That would be the goal, given the data that Professor Schett has published so far in other autoimmune diseases. And again, just as I described, previous studies have shown that a single dose of these CD19 CAR T cells using a similar 4-1BB CD3 zeta structure, which is the same structure that was used by Professor Schett, with these types of elements, with these elements in his molecule as well, is well tolerated and has led to deep and transient B cell depletion while inducing a significant clinical response in patients with other B cell mediated autoimmune diseases. These are lupus, idiopathic inflammatory myopathy, and systemic sclerosis. Therefore, a CD19 CAR T-cell treatment has the potential to offer new therapy to patients with myasthenia gravis that can mitigate the need for long-term immunosuppression. And so just to describe briefly what's called the RESET myasthenia gravis trial, that is Cabaletta's phase 1/2 clinical trial of CABA-201 in generalized myasthenia gravis. There are 2 cohorts that will enroll in parallel: 6 patients with AChR positive disease and 6 patients with AChR negative disease. That would be patients who are MuSK positive, LRP4 positive, or seronegative. Our approach is that using the initial dose of CABA-201, which is equivalent to the dose used in the paper that I just showed you from Professor Schett regarding the same CAR T-cell, a similar CAR T-cell structure that is very similar to CABA-201, and that which has been evaluated in lupus, myositis, and systemic sclerosis. Some key inclusion criteria, remember I talked about these criteria that define the patient population that would be eligible for the study. These include patients between ages 18 and 70 inclusive, a diagnosis of generalized myasthenia gravis, and disease activity despite standard of care therapies. Key exclusion criteria, remember these are the criteria that if a patient had this, that they would be not eligible for the study. This includes myasthenia gravis with only ocular manifestations, an active or untreated thymoma, and treatment with a B cell depleting agent within 6 months. So I'm going to go back to some poll questions, and I know that there may be different groups of attendees for our meeting today. So the first poll question is for patients who may be on the webinar today. So given what you've learned so far about CAR T-cell therapy in autoimmune disease, would you consider discussing participation in a study for CAR T-cell therapy for myasthenia gravis with your physician? Just going to let another few seconds go by here until we have responses. All right, so it looks like the responses have stopped coming in. I think we can end the poll. And the results are that 88% of respondents said yes, and 13% of respondents said no. So we'll follow up on these questions in just a bit. I'm now going to switch to any physicians or providers who may be on the webinar. Given what you have learned so far about CAR T-cell therapy and autoimmune disease, would you consider discussing a study for CAR T-cell therapy with your patients with refractory myasthenia gravis? I'll give a few seconds here for any physicians or medical providers or researchers to answer. All right, it looks like the poll is, I can close the poll out and I can share the results. So it looks like we had 5 respondents and 80% or 4 out of 5 said yes, and 1 out of 5, 20% said no. So we'll continue to discuss this a little bit, and then we'll come back around to additional questions in just a few slides. So again, just to reiterate that CABA-201 are manufactured from a patient's own cells, and that manufacturing process is similar to that used for CAR T cells used for patients with blood cancers, as I described previously. So again, this is a bit redundant with what I said before, but basically, again, the patients undergo a procedure called leukapheresis, where the white blood cells, including the T cells, are collected from the body. The T cells are then changed in a laboratory to insert the chimeric antigen receptor to target CD19. The changed T cells will be increased in number and then infused intravenously back into the body. And our CABA-201 clinical trial for myasthenia gravis is expected to be started this year in 2024. I do want to go back and talk about safety, because I think safety is obviously the most important thing for our patients, particularly in phase 1 and phase 1/2 trials, where you're getting that initial data to make sure that it is safe for patients to receive. Safety is always first. So I do want to go back in and discuss safety a little bit further based on what's been described and observed in patients with blood cancers, as well as for what's been observed in these patients with autoimmune diseases. So this is a slide depicting 2 of the main side effects or adverse events that have been observed that are under a lot of discussion and of great interest and importance to both patients, providers, and the companies that are studying CAR T-cell therapies, both in blood cancers and in patients with autoimmune disease. The first of those is called cytokine release syndrome. Cytokines are different signals that are present in some cells of the body. And some of these cytokines can cause a massive inflammatory response. And cytokine release syndrome occurs when the CAR T cells bind to their target. So they're binding to these B cells. And that leads to the release of a lot of cytokines as those B cells are killed. And those cytokines then make other immune cells release additional cytokines. And that can lead to a massive inflammatory response in the body that can manifest as fever. It can manifest as low blood pressure or hypotension, and it can affect organs like the lung and cause people to need oxygen. Needing oxygen is called hypoxia, low oxygen levels in the blood. And you can see here that the grading system for cytokine release syndrome is dependent on how much support a patient needs for their low blood pressure and or their low oxygen. The other major side effect, an adverse event that we worry about, is called immune effector cell associated neurotoxicity syndrome, or ICANS. The exact pathogenesis or pathway by which this is caused is not exactly clear, but it's felt to be very similar to cytokine release syndrome in terms of the risks that patients have in developing this. And this manifests as changes in consciousness, can result in seizures, changes in the ability to have motor neuron findings, motor muscular findings, meaning the ability to move, and even can lead to swelling of the brain and increased pressure in the brain. So these are not adverse events to be taken lightly. These adverse events, cytokine release syndrome and ICANS, have risk factors in some clinical trials in patients with blood cancers. Some of those include higher bone marrow tumor or malignancy burden. That makes sense because the amount of cytokines that are going to be released are going to depend on the amount of B cells that are being killed in the case of CD19 positive, CD19 CAR T cell therapy. And larger doses of CAR T cell therapies is also associated with higher risks of severe cytokine release syndrome and ICANS. Again, making sense that the more interaction you have between the T cells and the target cells, the more cytokines that are going to be released and the higher the risk. And remember that in patients with blood cancers, the thought is that the number of target cells is much, much greater. The number of B cells that are there in the patients with blood cancers is much, much greater than in patients with autoimmune disease. So the idea is that patients with autoimmune disease should have lower adverse events related to cytokine release syndrome and ICANS. That has been the hypothesis going into these trials for, or the educated guess going into these clinical trials for CAR T cells in autoimmune disease. And also the structure of the CAR T cell itself have aspects which are associated with different rates of these adverse events. As I mentioned, this 4-1BB component, which is what's in CABA-201, and which is what is also in the structure that Professor Schett used in Germany, has a lower rate of adverse events in some clinical trials in blood cancers compared to CAR T cells that contain this other domain called CD28. So all important considerations as we think about adopting these therapies into patients with autoimmune disease. If we go back to the data from Professor Schett's group, this is the most recent abstract that he presented on his 15 patients, which was presented at the December 2023 American Society of Hematology meeting. And again, this is 8 patients with lupus, 4 patients with systemic sclerosis, and 3 patients with that myositis, idiopathic inflammatory myopathy. And here again is the safety data. We have grade 0 cytokine release syndrome, which is nothing at all in 4 subjects. Grade 1 cytokine release syndrome, which is fever only in 10 subjects. And grade 2 cytokine release syndrome in 1 subject. This is a patient in the IIM group, the myopathy group, who had pre-existing lung disease and fever in the setting of cytokine release syndrome, who required oxygen, supplemental oxygen while the patient was febrile, but then that resolved. And 6 of the 15 patients received the medication that's used to treat cytokine release syndrome, which is called tocilizumab. This is standard of care. It's routinely used in the patients with blood cancers for cytokine release syndrome. With regards to that neurologic toxicity, 1 patient experienced grade 1 ICANS presenting as vertigo 2 weeks after infusion, and that resolved with a short course of steroid medications called dexamethasone. And no patient had prolonged bone marrow suppression, meaning that beyond a month, no patient had severe depression of the cells of their bone marrow, which make immune-related cells, which make red blood cells, which carry oxygen to tissues, and which make platelets, which help blood to clot. So that was not observed. There is a very important announcement that I do think it's important that we recognize, however, which was an announcement in November of 2023 from the Food and Drug Administration regarding the risk of T cell cancers after the approved CAR T cell therapies that are already on the market. So this is just a snapshot of the announcement on November 28th, 2023. And I'm going to read this verbatim because I do think it's important that we stick to the language that the FDA has put out about this, because there are many questions about this and a lot of discussions and articles about this, both in the scientific literature and the lay press. But I do think it's important to recognize the language that the FDA has used. So in that announcement, the FDA has received reports of T cell malignancies, including those that are positive for that chimeric antigen receptor, in patients who receive treatment with these BCMA or CD19-directed autologous CAR T cell immunotherapies. Reports were received from clinical trials and or post-marketing adverse event data sources. That means after the drug was already approved. The FDA has determined that the risk of these malignancies is applicable to all of the currently approved BCMA-directed and CD19-directed genetically modified autologous CAR T cell immunotherapies, and these T-cell malignancies have occurred in patients treated with several products in the class. Now, this last statement we did modify with an update because there was an update just a week or two ago, that although the benefits of these products continue to outweigh the potential risks of their approved uses, that's from the FDA. The FDA is continuing to monitor the identified risk of T-cell malignancy with serious outcomes, including hospitalization and death. And this is the update, and has requested that all of these approved CAR T-cell products update their prescribing information and medication guide to identify the possibility of the increased risk of getting cancers, including certain types of cancers of the immune system. So again, I want to be very transparent in our discussion today that we take this announcement very seriously at Cabaletta, that safety is the #1 priority for the patients that we would like to help with these CAR T-cell therapies, but are studying in these phase 1/2 trials. And I would like to say again that still there is a lot of information. If you Google around or you check on the internet, there are a lot of opinions being put out by experts in the field and centers that have treated thousands of patients with these CAR T-cell therapies. But it's important, I think, to stick to the language for this presentation as to what the FDA has said, so you can do your independent research and counseling with your providers and patients around these risks. Clearly, we still need a lot more information to understand the exact causality here and how many patients have actually developed these T-cell cancers after CAR T-cell therapy. So I know I staggered the questions here for a specific reason, because obviously I showed you the efficacy data, which was very promising, but we do want to be very transparent around the data that's out there. So given this additional information, would you consider discussing participating in a study for CAR T-cell therapy for myasthenia gravis with your physician? This is for patients. Great. I think I can end the poll here. Looks like still we're at about 83% of patients electing to discuss this with their physician if it was available, and 17% electing not to discuss. And for providers, physicians and providers, let's go on to the next poll question, which is 3B. Given this additional information, would you consider discussing a study for CAR T-cell therapy with your patients for refractory myasthenia gravis? We have, I think 5 people had responded before. We have 3 so far, and I think we're going once, going twice. And I'll end it there. And we have all 3 providers, patient providers or physicians, saying that they would consider discussing a study for CAR T-cell therapy with your patients with refractory myasthenia gravis. Obviously, all of this is with the most updated information that can be provided for counseling, for assessing the risk to patients, as well as the potential benefits as we think about whether this is appropriate to think about for patients with myasthenia gravis. And I'll finish with this last question. This is for everyone on the line today. What most interests you about CAR T-cell therapy for myasthenia gravis? This is poll question number 4. I don't know if, Dova, if we can get this last question up. Maybe we don't have it loaded up, but I guess we can answer in the chat if it doesn't pop up. What most interests you about CAR T-cell therapy for myasthenia gravis? Number 1, it's a one-time treatment. Number 2, the ability to stop all other medications that you would take for your myasthenia gravis. And number 3, the safety data generated from studies in patients with autoimmune diseases so far. If we don't have it for the poll, you can also put an other in there, since the chat, which Dova is moderating.
Yeah, I don't know why it's not coming up, because
that's okay.
Okay.
That's okay. But yeah, we can obviously have a write-in answer here, which is always great as well, to hear answers outside of what I've written down as these potential answers. And with that, I will stop. I would like to again I'm going to stop and take questions, but I do want to thank Dova, Meredith, the MGFA. I would like to thank all of the participants who have joined the webinar today, and all of those who will subsequently view it. And again, thank you so much for the opportunity to discuss clinical trial participation, CAR T-cell therapy, and specifically the CABA-201 program for patients with generalized myasthenia gravis.
Okay, great. Thank you so much, Dr. Hogan. Our first question is from a patient who says, excuse my ignorance, what B cell depleting agents would I be familiar with?
No, yeah, no, and we appreciate all questions. And believe me, this is not, I want to say this is not simple stuff, right? So it's hard enough to understand a complex disease like myasthenia gravis. The immune system is extraordinarily complicated. Then there's all these other drugs, and then you throw in CAR T cell therapy. So this is why we do this as a team, right? This is a team effort to understand, to educate, to understand what are our potential risks and benefits here in thinking about any therapy. So the B cell depleting agent that is most well known is called rituximab or Rituxan. This is called a monoclonal antibody therapy, which targets B cells through a different protein called CD20, and that's the most well known anti B cell therapy that is used for autoimmune diseases. There are others that are used for autoimmune diseases and B cell cancers. But Rituxan would be the most well-known. Rituxan is the trade name and rituximab is the generic name. That's given by infusion, usually given by infusion. The initial dose is either once a week for 4 weeks or once every 2 weeks for 2 doses. And then you may need, patients may need additional doses every 4 to 6 months afterwards.
Thank you. So we do have a patient on the line who on the webinar who is currently in the clinical trial, and she just wanted to say that she's doing great, and it's been 5 months since her infusion. So she just wanted to throw that in there. So that's great.
Excellent. Thank you for that comment. Yes, I would like to clarify that the CABA-201 trial for generalized myasthenia gravis is not yet enrolling. We do have a study, which is our MuSK chimeric autoantibody receptor T cell therapy, which I did not mention because it is already enrolling, and I did not want to give the impression of patient recruitment. But that study is in the public domain on clinicaltrials.gov. And there is also another chimeric antigen receptor T cell trial that is being performed with another company. And so again, just to be clear for the audience around what are the potential CAR T cell therapy trials that could be relevant for patients if reporting any outcomes.
The next question is, the results sound incredibly promising. Has there been any hypothesis on what the success rate would look like for generalized MG?
Yeah, thank you very much for that question. So obviously we agree that the results look very, very promising for in patients with autoimmune disease. I can stop sharing here, so I can come on screen here. I don't think it would be appropriate to hypothesize the response rate in generalized myasthenia gravis because we do not have any data yet in that disease, right? I think as I outlined our study rationale, we think that we have good scientific rationale for targeting B cells with a CD19 directed CAR T cell therapy that has a 4-1BB co-stimulatory domain. However, I think it would be premature to give a prediction about what we would think would be the response rate.
Okay, thank you. Are there any trials that are planned to include subjects over 70?
Very good question. So as I mentioned, our initial inclusion-exclusion criteria is up to age 70. I think what we tried to do in our phase 1/2 trials is to obtain, again, safety is first. And so obtaining safety data in our initial inclusion-exclusion criteria would be the initial plan. There's always opportunity to adjust that later on, whether it be any of the inclusion-exclusion criteria. Right now, these are our inclusion-exclusion criteria. I cannot speak to any of the inclusion-exclusion criteria in other CAR T cell trials for myasthenia gravis. But obviously, we understand that in all of the diseases for which we are studying CABA-201, that there are patients that are impacted outside of the ages and other inclusion-exclusion criteria. So I think that would be an opportunity for further exploration for any therapy by modifying inclusion-exclusion criteria, including the age range, after the initial safety and efficacy data has been obtained.
And if everything went the way you thought, would this be also be used? Someone's asking, would this impact seronegative as well as seropositive patients?
Well, I can speak that in our trial, we are, as I had mentioned in the in one of my slides, that we do have AChR negative patients included in the trial in one of the cohorts, and so correct, there would be an eligibility, correct. So we would get the data for AChR positive and AChR negative patients, and so all of those subgroups would be included in the analysis, and then based on the safety and efficacy that are observed in the phase 1/2 trial, then decisions would be made about further programmatic planning around how to best explore this in the different patient cohorts with myasthenia gravis.
Great, thank you. So as we know, there's numerous new medications that are approved for MG. How will this therapy fit into the treatment line? We have first-line therapies, second-line therapies. When do you see doctors approaching patients with the conversation of this treatment?
Yeah, so this is an excellent question. And actually, this is a question that's excellent both for patients and providers and any physicians who would be thinking about participating in such a clinical trial. So designing the inclusion-exclusion criteria for a first time we're studying a drug in humans is very important, right? Because again, safety is first. So as I mentioned in the inclusion and exclusion criteria, that patients will need to have failed some therapies prior to considering and being eligible for the CABA-201 trial. And again, I think that that's a reasonable approach because we do have, as you mentioned, many therapies and which have been approved in the last 5 years or so, right? Many therapies have been approved recently. And so this is great news for the myasthenia gravis community that there are all these approved therapies. So I think that in the initial study, there will be the inclusion-exclusion criteria around specifically having failed a certain therapies prior to undergoing this treatment in the setting of the clinical trial. And then depending on the safety and efficacy of the therapies, then decisions could be made about which patient populations it would be appropriate to study subsequently. And again, I can't speak to exact plans in myasthenia gravis, but what we do know is that in the setting of hematologic malignancies or blood cancers, that these are studied in a specific population to start. And if the drug is safe and effective, then obviously one could consider studying it earlier on in the therapy of patients with any disease. So again, I can't speak to exactly where it would wind up. That will be based on the data and the clinical practice guidelines that would subsequently come out and the package labeling for any therapy. But I will say that that there is precedent, obviously, with many therapies in the beginning of getting good data on safety and efficacy, and then it being applied to different patient populations than for which it was originally studied.
So I'm going to ask one last question. There's a few other ones that we've kind of gone over before, but this one I'm going to ask one last question and we'll be done. Clarification, please. Is this a one-time treatment, or will there be follow-up treatments needed? I think people just can't believe it. So I just wanted to ask that and let you speak to it again.
Yeah, and it is unbelievable. I think that's the point, actually, and that's why I told the stories I did about patients with these blood cancers, right? Because in these patients with blood cancers, it was a one-time therapy. There are some patients in the literature with blood cancers who have been retreated, but that is very rare. And so the idea, and all of the patients in the autoimmune disease setting that I described from the group in Germany, this was a one-time treatment. So again, I want to reiterate that the approach here is that patients would receive one infusion, and then again, based on the precedent of the autoimmune disease papers so far, that their approach was to take patients off or taper them off all of their other therapies subsequently. And that was that drug-free response that I described. So again, to reiterate, this is a one-time treatment. And that is what separates this type of therapy from other therapies. Obviously, there's a lot of enthusiasm. That's why I wanted to present, hopefully, a very balanced webinar today by showing both the really exciting efficacy data and also, comment specifically on the safety data that I presented today. But that's why there is excitement around this type of therapy that is unique in the field of autoimmune disease compared to almost any other therapy, because it is a one and done therapy. And that is a very unique and very transformative in the approach for patients with autoimmune disease. So again, I think we want to be very cautiously optimistic. The data will be coming in, obviously, as we get more data for patients in clinical trials. But this is why when we have that mission statement about providing a curative targeted cellular therapy for patients with autoimmune diseases, the precedent has been set with some patients with blood cancers. And so that is our goal, is to try to recapitulate that experience for some patients with autoimmune diseases.
So in closing, I don't normally read the comments in the questions. I usually read the questions, but there is one comment that I think you would like to hear. And she says, "We are parents and our daughter has MG, and you are giving us hope." So with that, that's amazing. So I want to say thank you, Dr. Hogan, and we appreciate your time today. And this is truly exciting.
Thank you very much. Thank you, Dova. I appreciate it. And just to speak to that last comment, again, we do appreciate that feedback. That's what keeps us going, right? That is our motivation. Our motivation truly is to cure or get as close to cure as possible for our patients with autoimmune diseases. So that gives us good energy to keep on pressing forward. So thank you so much for that. And we really do appreciate the opportunity to have this webinar and discussion today.
Okay, thank you, everyone. Bye-bye.
Thank you. Bye.